Hello, and welcome to this webinar hosted by Jen. Today's topic is Mapping the Invisible, Building the Largest Database of Human Microbiomes. Today's webinar is sponsored by MGI. Thank you so much for being here. I'm Uduak Thomas, Senior Editor with Jen, and it is my pleasure to host today's webinar. We have two speakers lined up for you today. Dr. Matthew Almeida is a research fellow at INRAE Metagenomics Institute. We also have Dr. Micah Seifert, Head of Unit at the Center for Translational Microbiome Research, National Pandemic Center at the Karolinska Institute. Before we get to their presentations, let's get some background on the Million Microbiome of Humans project. This is a 5-year project that officially launched in 2019. Scientists from 21 institutions and more than 10 participating nations are cooperating to sequence and analyze 1 million microbial samples from the intestines, mouth, skin, reproductive tract, and other organs. The ultimate goal of the project is to draw a microbiome map of the human body and improve scientists' understanding of the functional capacity of the human microbiome, as well as of human health and disease overall. To date, nearly 130,000 samples have been sequenced. When it is complete, it will be the largest and most comprehensive human microbiome database to date. In this Gen webinar, our experts will share their experiences with using microbial metagenomic sequencing to build the database. They'll cover the technologies and protocols used to collect and analyze data on microbial communities, as well as to assemble and characterize the genomes of novel species. Before we begin, a reminder that we'd love to take your questions following our main presentations. At any time during the presentations, you can enter your questions in the Ask a Question panel located on your screen. All right, let's get started.
Hi, it's a pleasure to welcome you to my talk, Unveiling the Microbiome Mysteries, Navigating Challenges and Revolutionizing Human Healthcare with Next-Generation Sequencing in Microbiology. I'm Micah. I'm the Head of Unit of the Lab Core Unit at the Center for Translational Microbiome Research at Karolinska Institute in Stockholm, and I'm the CEO of Embiogen, a newly founded startup company that provides metagenome sequencing services. I'm going to start with explaining to you a bit about what the Center of Translational Microbiome Research does. So we focus mainly on better understanding how the human microbiome is involved in different health and disease states, with the goal to open up opportunities for the development of novel therapies in the area of gastroenterology, reproductive health, and cancer. We have been founded 2016 as a collaboration between Ferring Pharmaceuticals and Karolinska Institute, and have since then established a broad technological platform to study different complex microbiological communities in well-defined human materials. The platform includes today a biobank of approximately 200,000 human samples with clinical metadata and 1,500 bacterial isolates with phenotypical and genotypical screening pipelines for the definition of consortia aimed for biopharmaceutical drug development. As I mentioned previously, we aim to better understand the microbiota involvement in different health and disease states in gastroenterology, cancer, and women's health. But in order to understand the involvement of the bacteria in different health outcomes, you need to understand how the microbiome in a healthy community looks. So our center research evolves around the definition of a healthy microbiome. And while we are better and better understanding how a normal microbiome looks and how the microbiome influences different health and disease states, we are hoping to long-term change our view on therapeutical practices. We see that there are lots of opportunities for therapeutic intervention and prevention, how microbiome components can be used as biomarkers to define certain disease states. We can see that microbiome components can have a direct impact on the human physiology and pathophysiology, and how the modulation of the microbiome can change certain health and disease states. We see that in the future, precision medicine will include the microbiome. So I'm going to talk you a bit through how we are doing this. We focus mostly on metagenomics and culture studies in our center. We perform metagenomic sequencing to define the community, to do taxonomic profiling in different human tissues, but we also look at proposed functional studies. So we look at the different genes that the bacteria in a sample do possess and which proposed functions they could have in the tissue that could interact with the host. And we do culture studies where we isolate bacteria and do functional assays and them in phenotypic and phylotypic profiling. We have in the recent years built a quite substantial platform for metagenome sequencing. The most important thing when you do big population-based studies is to have a standardized platform. You want to include as little bias into your results as possible, which means that you treat all the samples in exactly the same way. In our center, we try to take control of as much of a study as we can. So we help our collaborators to plan the study and the study material. We have a laboratory tracking system that tracks all the materials that go out of the center and that go into the center, from freezer storage through the whole pipeline to the data storage in the end. So in a typical sample would be placed in sample tubes provided by us and then sent back to us. The sample tubes are normally labeled in a three-way labeling manner, which means that they have three different sets of barcodes which can be used for identification. Then the sample, once a study is concluded, a sample will be entering the screening pipeline in a fully automated DNA extraction, where all samples are extracted in a defined way, exactly the same way, with a plethora of controls. And from then on, they will go further into library preparation, where a metagenome sequencing library will be prepared that then is sequenced on different sequencing platforms, either mid or ultra high throughput platforms, from where the data is automatically uploaded to our storage systems and then automatically processed. Further analyses are then done by our bioinformaticians. Today, our platform has a capacity of approximately 1,500 samples a week. And this is important because in order to do big population-based studies to define how a normal microbiome looks and to handle them in a as little biased as possible way, you need quite a big throughput. There is a lot of noise in human data in what's normal. We all have a unique microbiota profile, and in order to find those components that are similar and that do have an influence in a bigger part of the population in different disease and health states, you need a very big population-based study. Since we are in a part of technological development where changes are quite frequent, we need to process those samples as close in time as possible. Big studies processed in a close time means high throughput. So we see that we are only able to provide the data quality that we are providing today with the capacity that we have today. We do also a quite extensive development in our center in order to standardly process samples. We have optimized extraction and library preparation procedures for quite many sample types. The sample types that mostly go through our pipelines are fecal samples, vaginal swabs, biopsies from skin and intestinal tract, saliva samples, gum cavity samples, and skin swabs. For all those sample types, we have standardized pipelines. So all those sample types, when they come into our center, they will be treated exactly the same way, which consequently leads to that you can compare those samples in a very precise way. There are no minimum input requirements anymore for sample types that normally have very little bacterial content. We do not require minimum input anymore. We can, if there are bacteria there, we can pick them up. We are today able to do direct quantification of bacterial mass in all sample types with the help of spike-ins in both extraction and library preparation that are used to exactly quantify every bacteria in a sample. We have targeted multiplex amplicon panels for different sample types. And we do quite intense development on metatranscriptomics. One of our biggest development projects as of today, the one that I'm personally most proud of, is the host depletion for sample types where you have quite massive amounts of host DNA, like a biopsy or skin swabs. Even vaginal swabs have quite high amounts of human host content. You do sequence today mostly those human contents. So for a stool sample, you normally see very little human DNA. So most of the DNA that you extract from the sample will be relevant for your research. However, if you would be processing a vaginal swab or even a biopsy sample, between 60 and 99% of the DNA you will be extracting from the sample will be of human content. So you will be using 60% of your sequencing capacity on sequencing information that you're ultimately not interested in. We see this as a big waste. So we want to focus on the microbial communities and consequently reduce the costs for sequencing, data storage, and bioinformatic analysis. Early data in our development shows that we can reduce up to 50% of the host reads depending on the sample types, and that host removal will be a breakthrough for boosting sensitivity to characterize rare and less abundant microbial contents. This means that where you maybe had 1% of bacterial content in a biopsy sample, that you will be able to sequence at least double as much data since the community diversity is very big in those samples. You will be able to pick up much more of those less abundant samples, which in the end will give you a much bigger insight, because those are the bacteria that we see are most often most influential on health and disease states. In the plots you can see on the bottom of the slide, you can see that there is in almost all samples increase in knowledge. So we can see that there is an increase of bacteria that we are able to detect, which shows us that deeper sequencing is indeed relevant for those low abundant species. So now that you've seen how our pipelines work and how we work, I want to talk to you a bit about what we are doing. So as I said in the beginning, we focus on the microbiota involvement in different health and disease states in different target areas. And one of them is gastroenterology. And there we are looking into how different microbiota profiles influence, in particular, IBD. So we want to see if there is a different microbiota profile in healthy patients compared to diseased patients. We want to see if there is a microenvironmental part to this. So if in irritable bowel syndrome, diverticulosis, different polyps and tumors, if those tissues are differently colonized than normal tissues, if this is different in children than it is in adults, if we can ultimately identify biomarkers that would improve the diagnosis for IBD, which up to date is still very complicated, and if we maybe even can change the microbiome into a healthy profile from a diseased profile. We do this with longitudinal studies. In Sweden, we have the extremely great opportunity that all health data is stored with a unique identifier for every person. So we have very big longitudinal studies where people leave in regular intervals fecal samples that we can then track together with the National Cancer Registry data for inpatient registries. So those samples have been collected over many years from different individuals. We have up to 60,000 microbiota samples today. And those samples we do taxonomic profiling on, so metagenome sequencing, and then track the results, connect the results to the national registry. So we know which patients are healthy and have stayed healthy over the time of the study, and which patients have developed a diseased state, so have been diagnosed with either IVD, IBD, diverticulosis, have developed polyps or tumors, and can then identify, hopefully, biomarkers that can help to diagnose those cases much earlier than we can today. Another of our main research areas is women's health, and there our main research questions are if the microbiome acquisition already starts in utero, if the menstrual cycle and hormonal contraceptives have an influence on the microbiome, what a normal microbiome looks in a healthy woman, and if there is a profile that can be associated to pregnancy complications, especially if we can identify patients that are at risk for preterm birth or pregnancy loss. And then finally, if there is microbiome involvement in the endometriosis and fertility. Here I would like to highlight one of our biggest projects, the Swedish Maternal Microbiome Project, or SWMAMI for short. It includes 5,000 mothers and 3,000 babies. And the main aim of the project is to assess the association between the maternal microbiome during pregnancy and postpartum and the risk for maternal and neonatal adverse events. With the first main research question, if there is a involvement of the microbiome or a specific microbiome profile that is associated to an increased risk of preterm birth or pregnancy loss. I'm going to talk you a bit through how we collected the data. So we collected data from the mothers 20 weeks into the pregnancy and between week 28 and 30 with an additional questionnaire, and then 5 weeks postpartum also with a questionnaire. The samples we took are fecal samples, vaginal samples, and saliva samples from the mother, and the postpartum sample includes a fecal sample from the baby. The taxonomic profiling results are then also registered, as in a previous study, to the National Health Register and evaluated together with the questionnaire data to assess a normal and healthy microbiome in a normal pregnant woman and a normal baby or a healthy baby, but also look into different pregnancy complications and health complications and a potential involvement of the microbiome into those health states. As I mentioned previously, in this study we have samples from over 5,000 mothers and 3,000 children that were born between 2018 and 2021 in Sweden, which gives us about 50,000 samples in this study. And luckily, a lot of those pregnancies went very normal with healthy mothers and healthy children. But we also do have a substantial amount of pregnancy complications, both diagnosed and self-reported. And those pregnancy complications are so extensive that they give us the possibility that together with all the healthy participants, we have a very unique sample set that enables us to define biomarkers and define microbiome states that are correlated to those states, and hopefully give us the chance to develop targeted therapeutics to prevent or at least early detect those states. Another project that I would like to highlight is the Million Microbiome of Human project. It's a collaboration between different laboratories in Latvia, France, Denmark, and here in Stockholm, together with BGI Research and labs in China, where Our aim is to map the normal microbiome in order to be able to facilitate those studies better. If you're interested in the Million Microbiome of Human Research project and the Microbiome Atlas, then you can scan the QR code if you want to get in contact and be part of the project. You can scan the other barcode and get in contact with Binyan. It's a huge project that will hopefully enable us to better understand how a normal microbiome looks and facilitate the interaction between different researchers in the world to contribute to the field with more standardized data that give us the possibility to get a better understanding of microbiomes in different ethnicities, with different cultural backgrounds, and different disease and health states. As a company, we would like to enable you to get the best out of your metagenome study as well, with cutting-edge sequencing technology and cutting-edge method development, enabling you to have the state-of-the-art metagenome sequencing results that your project deserves. We have more than 10 years of experience in this field. As I mentioned earlier in my talk, we have in our research fields spent a lot of time in developing the best metagenome platform that you can imagine. And now we would like to offer this to customers outside of the research field as well, or also outside of the research collaborations that we have. We are able to supply you with tailored workflows that meet your unique requirements for your project. If you're interested in this, you can scan this barcode and get in contact with me so we can talk through your project and the possibilities for it. And finally, I would like to thank my team, Yenghua, Alex, Bianca, Ahmed, Yeva, Pinal, Tao, and Philip, who make all of this work possible, who work very hard in the lab and the development, and who have been with me for a very long time. They're the best team I could wish for. And the Center for Translational Microbiome Research, that is more than just metagenome sequencing. All of those are the people that take part in the study designs, that take care of the samples, that take care of the shipment for the materials and how to get the materials back and how everything stays controlled in the freezers. And finally also gets analyzed and take care of our extensive clusters and the health of our technology platform. So thank you to them for making this great work possible, and thank you to you for listening.
Hi everybody, my name is Mathieu Almeida. I'm a researcher at INRAE Jouy-en-Josas, and today for this webinar I'm gonna talk about the human digestive microbiota and its exploration from the pioneer MetaHit project to the Million Microbiome of Humans project, which is a new ambitious project that aims to analyze 1 million microbiome worldwide. So the humans are covered by microorganisms from the skin to the mouth and inside our body, different compartments of the human digestive tract. And if, for instance, you analyze a fecal sample of the human microbiota, you will find different types of microorganisms, bacteria in dominance, but you will also find viruses associated, and also you could detect sometimes archaea and other organisms like eukaryota, protozoa, and fungi. And also, depending on the host's health status, you can also detect human DNA in a high or low concentration, depending on the ecosystem. And so, when analyzing the specific microbiota, it's important to have a look at all the different types of microorganisms and their potential function associated that could be linked, that could be a clue, consequence, or participate in the human health. So to analyze a specific microbiota sample, you can use the metagenomic sequencing technique. So it's a powerful tool that allows to explore the link between the microorganisms and a specific phenotype. And for performing an efficient metagenomic analysis, Each steps are very important, from the collect to the transfer of sample, then the DNA extraction, trying to extract all the DNA from the different type of microorganisms that you can find. Then the metagenomic sequencing with sufficient depths of sequencing that will allow you to perform taxonomical and functional exploration. Then the taxonomy and function exploration using an appropriate and representative human microbiome reference database. And at the end, performing relevant bioinformatic and biostatistic analysis with normalization procedures, for instance, that will allow you to make the comparison between different types of phenotypes and detect biomarkers of the specific disease. So for that, conventionally, when we perform metagenomic analysis, we have to deal with millions of short reads, usually 150 nucleotides of length, when you perform short read sequencing. And so these millions of reads, you can either do de novo assembly on them, or you can directly perform a reference-based analysis, which is faster because you have the genomes, the genes, and the functions annotated. And then directly you can align these reads to the reference that you analyze. You will be able not only to detect the genes and the function, but also have the different abundance between the different specific features that you will detect. And at the end, you will be able to perform genomes, and function exploration. For the human gut, you have different type of database. So the database is something that allows you to explore in depth a specific ecosystem. This started in 2010 by the MetaHit project, which was a pioneer project which created the first human microbial gene catalog of the human intestinal microbiota, using 100 fecal samples from Europe and China. Then, with the time, new fecal samples were published and available for the community, and the database and reference catalog was updated accordingly. So, new American samples were sequenced and available. And then, from this day to today, we can today find fecal samples and human microbiome metagenomic samples from all over the world. And the important question for you when you analyze a specific ecosystem is to know if the database you are using is representative of the ecosystem you're analyzing, and also specifically to the phenotype you analyze. So in the MetaHit project, not only they provided a reference catalog, but they also provided new a concept of analysis. So for instance, here is the enterotype concept. So in 2010, they introduced this enterotype concept, which is a gut microbial structure, which is independent from nation, age, gender, or BMI. So at that date, they observed that the different microorganisms that you can find could be dominated by different type of microorganisms, and using different clustering technique, they were able to cluster in three type of sample. The samples that are Bacteroides dominant, the one that are Prebola dominant, or Ruminococcus dominant. So here is a PCA plot, a representation of this clustering, and each dot is a sample. And as you can see, you have a different type of sample. Some of them that are closely related, and they have this taxonomy dominance of Bacteroides here, for instance, or Ruminococcus or Prebotella. And what they found is also that some clusters seems to be enriched in specific type of individuals. For instance, the Bacteroides, they had a little more IBD related individuals, and the Ruminococcus dominant had a little more healthy related individuals. So more samples give you more new microbial structure to explore. And for instance, in 2020, the MetaHit CARDIS cohort provided about 1,000 European fecal samples to analyze, performing shotgun metagenomic. And what they found is, with this PCA plot, is that as before, they observed the different enterotypes that were analyzed, and inside the Bacteroides enterotype, they observed a structure inside of samples that were dominated by Bacteroides, but with very low Faecalibacterium species abundance, Faecalibacterium genus, which includes Faecalibacterium protenici, which is known to be anti-inflammatory. And so they observed that this specific subpopulation was associated with more metabolic syndrome disease, higher obesity, and that could actually be a structure of microorganisms where you are in a higher inflammation, potentially higher risk of metabolic syndrome disease development. So with new samples included in the analysis, you can find new microbial structure. And the idea is to actually increase the number of samples we analyze to discover all these new microbial structure, and then identify the association between the microorganisms conform structure and a specific phenotype. And so for that, at INRAE, we performed a worldwide large-scale analysis, including all the samples that were analyzed from the Metacardies, the published one, and all the other samples with different phenotype associated. And we use specific bioinformatic and biostatistic techniques to normalize all the samples and so make all the comparison and make this big database called the Human Microbiome Atlas in collaboration with the King's, the KCL and SciLab Lab in Sweden. And with this new database, we observed specific microorganisms that are depleted in disease, like the Akkermansia mucinifila, who seems to be depleted in a lot of disease. And we identify also organisms that are enriched in disease that potentially could be pro-inflammatory, like Streptococcus, some Streptococcus like Parasanguinis. And not only we can identify taxa that are associated or enriched or depleted in disease, but we can also find functions carried by these microorganisms that are depleted or enriched in specific disease. And what we saw in general is that the microorganisms that are depleted in disease, usually they lack vitamin synthesis and they are enriched in virulence factor. So that could actually induce pro-inflammatory health risk. But the problem with all these things is that often the clinical data are limited. You only have age, BMI, and sex, or even less than that, you don't have the sex or the BMI. And most of all, the samples were processed in different ways, different type of DNA extraction procedures with different type of metagenomic sequencing depths. And this could actually be a problem for interpretation. So let me give you an example of this kind of situation. Here is an example of paper from Thomas et al., which is a meta-analysis in colorectal cancer disease. In this meta-analysis, they collected different samples from different papers with individuals that have either colorectal cancer or an adenoma, which is a polyp that is detected and could actually be induce colorectal cancer, and individuals that are controlled, and they try to have these from controlled and colorectal cancer individuals for each of the study. And as you can see, in general, you have a little number of individuals that are analyzed. Sometimes the sex is missing. And at the end, it's difficult to have, sometimes for some specific cohort, a matching between age, sex, and BMI between the healthy and sick individuals. And because of that, you can have a misinterpretation. But still, this is an interesting meta-analysis with a lot of samples, and they performed the analysis. And what they did is they identified the organisms that are either enriched in colorectal cancer individuals or enriched in control individuals. And the number you see inside these boxes are the ranking of these species in AI model, AI prediction of colorectal risk assessment. If it's close to 1, it means that these microorganisms are often used to predict the risk of developing colorectal cancer. And if the number is higher, then it's not in the top list of species that are used to detect this disease. And then what they did is they did area under the curve analysis to predict the risk of developing colorectal cancer. When it's 0.5, it means that your prediction is not as good as flipping a coin, and you have one chance on 2 to actually make a mistake. And when it's close to 1, it means that you predict perfectly the risk of developing colorectal cancer. The different colors correspond to the different cohorts, and the number of features are the number of species that are introduced in the model to predict the risk of developing colorectal cancer. For instance, here I only use Peptostreptococcus tomatis and Streptococcus salivarius, or I use 4 species, 8, 16, 32, all of them. And what you can see is for almost all the cohorts, when you increase the number of species that you use to predict the risk of developing colorectal cancer, you increase the prediction of developing this disease, of having colorectal cancer. But for some cohorts, even if you include more microbial species, it's not improving actually the prediction of having colorectal cancer. And in this case, the reason was discovered by another team in 2022, is that this specific cohort not only had low sequencing depths, but also DNA quality was not as good as the other cohorts. And then this illustrates that if you don't have sufficient quality in sample collection and treatment, then you will not be able to have a good prediction of colorectal cancer in this context. So it's really important to masterize all the steps for the metagenomic analysis, because the bioinformatic will not be able to correct the problems sometimes if you have a low quality cohort. And so for that reason, it's really important to test the different protocols and define standards that give you actually the best metagenomic analysis. And this was actually performed in 2012 by the International Human Microbiome Standard, coordinated by Dr. Dushko Erlich and Dr. Joël Doré. They made this network to ask different labs around the world to test different DNA extraction methodologies and on the same fecal sample, and then ask the different labs to perform DNA extraction and send the extraction back to the lab. And then each techniques and labs are here represented as a number, the lab 1, 2, 3, 4, up until the lab 21, coming from 13 countries and 3 continents. And they analyze the DNA quantity that they have in the DNA extraction and the percentage of short DNA sequences. So this is also a quality parameter, meaning that if the DNA is too short, if all the sequences are too short, less than 1.8 kb, then it will not be sufficient for a high quality metagenomic analysis. So what you want is a technique that produce a high DNA quantity and a very low DNA fragmentation. And as you can see, for some of the techniques used in the different labs, you have techniques that produce sometimes relatively low DNA quantity, and some others that produce a high fragmentation, and even some techniques that induce both the two problems at the same time. So what you want is actually not necessarily to use the same standards for the different studies or the different labs, but what you want to know is if the different techniques used is efficient to give you a high concentration of DNA and a low DNA fragmentation. And that's the purpose of this network, is to provide guidance for the different techniques that exist to perform a high quality standard metagenomic analysis. So this was performed in 2012, but with the time, new techniques and new collection devices are commercialized. And that's why in 2021, this new network was created, Human Microbiome Action, new project coordinated by Dr. Joël Doré, to further improve the standards that we have today on these specific samples. Another way to have this huge analysis standardized is to have a large cohort created, and that's what we are doing in our lab at INRAE. We are creating a new cohort called the French Gut, and, which is also a consortium of public and private companies and institutes, with the aim is to collect 100,000 fecal samples from French donors up until 2027. And with the fecal samples, we also collect clinical and nutritional data, with the idea is to better define the heterogeneity of the human microbiota, intestinal microbiota, and also identify clinical and nutritional factors that could influence the gut microbial composition in health and disease. So the French Gut Project is open for any French donors in France. Today it's open for adults starting from 18 years old. It's also focusing first on the metropolitan France, but will then after to other territories of France. The idea is that each donors, they could freely connect online to a secured website, provide the eligibility and the questionnaires of nutrition and clinical information. When they fill all this questionnaire, they receive at home a specific collection kit device, which then provide all the information about how to collect the fecal sample. The fecal sample then is collected, is sent back to a APHP clinic, which we are working with. They pseudonymize the sample, and then they ship back to INRAE, where we perform the DNA extraction, metagenomic sequencing, and analysis, bioinformatic and biostatistic analysis. And then at the end, instead of doing individual feedback, which is forbidden in our context, we are performing a collective feedback for the donors, saying, for instance, that the vegan, they have this potentially this kind of signature, or this individual with this specific disease, we observe a specific signature when we have sufficient number of samples to give that feedback. So the French Gut data is actually operated using shotgun metagenomic sequencing. So we are using the NBSeq G400 technology, with the goal is to produce at least 20 million reads per sample. And we also collect health and nutritional data. We collect the million microbiome of human project metadata, minimal metadata set, which is actually the data that is collected for the different cohorts around the world that participate. So the French Gut is included in the million microbiome. And we also collect different metadata on top of that, 60 for the beginning of the project, but then we will include new nutritional, health, lifestyle, and diet questions depending on the disease also we will analyze. And the cool thing with this project is also connected to the health data system in France, which gives us access to 3,000 supplementary variables with a maximal historical depth of 20 years, meaning that we can have for each participant the information of the type of medication they used in the past and in the future. So we will be also able to know if they develop a certain type of disease, for instance, and then for this kind of individuals, look at the microbiome and see if by looking at the microbiome we were able to know if they were at risk of developing certain disease. To this day, we started the French Gut Project it was launched the pilot phase in 2022, and to this day we were able to collect more than 4,000 fecal samples. 12,000 donors are waiting to receive their kits, and we are planning to, with the Million Microbiome Consortium, to actually increase the number of participants we'll be able to analyze each year, with a maximum goal of 30,000 sample treated and analyzed each year until 2027. This is the kind of composition that we have in our cohorts. The bars that you can see on the left figure are corresponding to the different age. The large boxes are the French population, men and women that we expect. And as you can see, we are collecting a majority of individuals that are between 40 to 60 years old. And for some category, each age category, we are representative of the French population, and some we are lacking some individuals. For instance, young women or old men. And the idea is to then improve the communication in the future to collect more individuals for each categories. And as you can see, this is the population that in percentage on the right table that we expect, and we were able to collect almost the same number of the percentage per regions in France. So the idea of these large cohorts is to be really representative of the population that we will analyze. And on top of that, we have specific ancillary project inside the French gut with this study of colorectal cancer that we are starting in the European Microbiome project with the Professor Robert Benamouzik, who is the main gastroenterologist of the project in France. And we have a network of clinical centers that will participate at recruiting individuals with or without the colorectal cancer, and to know if the microbiome is actually a way to improve the colorectal risk assessment. So the French gut, as I mentioned, is part of the Million Microbiome project. This is a worldwide project that includes different countries and labs. With the core labs today includes the Sweden, Denmark, and France, and we have the support from China, which was also a founding member of the Million Microbiome, and Latvia. With the idea of this Million Microbiome project is to collect and analyze 1 million microbial sample from different ecosystems, not only the intestinal tract, but also the mouth, the skin, and reproductive tract. It's to draw a map of the microbiome using the best standards for microbiome analysis and generate the world's largest database. To, at the end, explore the link between the microbiome and health and potentially include this type of analysis for a national diagnostic for specific disease like colorectal cancer, as I mentioned. So the Million Microbiome is actually using high-throughput lab systems from MGI, which support the Million Microbiome. It includes different robots from aliquoting steps, DNA extraction and purification, metagenomic sequencing. And this pipeline can handle 96 samples per day, which allows us to actually perform a large-scale analysis each year, which is definitely what we need for the large cohort studies, trying to increase the number of samples analyzed and have a specific analysis using really robust standards. So this Million Microbiome project was launched in 2019, and to this day, 11 countries participate, which represents more than 23 institutes. More than 130,000 samples were collected and analyzed from more than 10 national cohorts, like the French gut. And the Million Microbiome is not a closed network. It's actually open for collaboration, and each participants that would be interested in participating could actually be included in the Million Microbiome and participate in this large-scale analysis. So here is the end of my talk. I would like to thank first all the INRAE team that participate for this French gut analysis. As you can see, there are a lot of people, which includes the people that collect, analyze, treat the sample, perform bioinformatic biostatistic analysis, as well as the communication ethics associated, the clinicians from the PHP that participate in the project, and the Million Microbiome that supports the project with Dushko Ehrlich in France, that is one of the founding members of this network. And as I mentioned, for the Million Microbiome, it's an open consortium. You can actually participate, and if you would like to be part of this big adventure, you can contact Binyam Okube, which is the MMHP coordinator, and see with him how you can be included in this big initiative. So thank you everybody for listening to this webinar. I hope you had a view of what the large cohort studies are bringing now, from the pioneer studies to these new studies with very large cohort. And I would like specifically to thank the Gen webinar members, with Dada Zong, Linda Kouvlar, and all the other members that gave me this opportunity to give this talk today for you. And all the INRAE, APHP, and MMHP consortium that are today giving new standards and new ways to explore the microbiome in the best way. Thank you very much.
Thank you once again for tuning in today, and thank you to our speakers for their superb presentations. In a few moments, we're going to move into our Q&A session. So if you haven't done so already, please submit your questions for our speakers now. Before the Q&A begins, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep your eye on your email and share the link with your colleagues who might not have been able to tune in today. All right, it's time for your questions. Just give us a couple of moments to transition. We'll see you shortly. Everyone, thank you so much for being here. I'm here with Micah. We're going to be taking questions live. A quick reminder that if you do have a question, now is the time to go ahead and enter your question into the Ask a Question panel on your screen. We're going to be taking questions live for the next several minutes, so please go ahead and ask those questions now. Hi, Micah. Thank you so much for being here, and thanks so much for that great presentation.
Thanks for having me.
Absolutely, absolutely. So we've had some great questions coming in throughout. I'm just going to go ahead and get started. This first question is actually region specific, but I think it probably is going to apply to other places as well. So the question I have is, do you have participants from Uganda? And this individual would like to participate, if you don't, would like to participate in collecting samples from that part of the world. So I think we can broaden that out if there are, I imagine it's a similar process, but I'll let you respond.
It's actually a very relevant question. Participants from Africa and Southern America are a bit underrepresented in the Million Microbiome of Human project. So we very gladly take participants from those countries because, as I mentioned before, the idea is to create a map of the human microbiome over the whole world. And for this, to be able to create this, we obviously need to have participants from all countries. So if you want to participate, I can only refer again to get information from our webpage, from the Million Microbiome of Human project, get in contact with Binyam, who's also, I think, present, and he will give you more information of what is needed in order to participate, what all the rules and regulations and guidelines are, and how to get in contact and how to apply to be part of the project.
Wonderful, wonderful. Thank you. All right, the next question I have, given its heterogeneity, how would you define the normal microbiome?
Also very relevant questions, very smart. It's absolutely correct. There's a huge amount of okay, I leave this out, in the human microbiome. And in order to define what's normal and what's not normal, you need huge population-based studies. And this is also getting back to the Million Microbiome of Human project. You need those significantly bigger studies in order to filter out the divergence in the data, because there is a massive influence coming from ethnicity, coming from your environment, coming from everything that is making you and you an individual. And in order to define the span in which normal lies, you need a lot of data, and to create as much as possible unbiased data, you need high throughput labs that are able to process those huge sample amounts.
Excellent. Thank you so much. And to our audience, I see those questions coming in. Thank you so much. As always, if you do have a question, please go ahead and enter it at any time. We're taking questions live right now. Next question I have, is this data available for public use?
The data is not yet available for public use, and there will be a timeline in which only participating parties are able to have access to the data. In the long term, however, the plan is to make this data publicly available.
And so this next question also asks about data, but I'm going to focus on the second part because it asks about the pipeline. So is the computational pipeline publicly available?
Ooh, that question I can actually not answer. I do believe so. But it's especially for the one that we are using at the Center for Translational Microbiome Research that is publicly available on GitHub. It's called STAG. It's from our own group. The one that is used at the Million Microbiome for Human project, I think is also publicly available, is however produced by a different group. It's not produced in Stockholm, it's produced in Copenhagen. And I would also refer there to go to the homepage and check that out and get in contact with the stakeholders in Copenhagen if you're interested in which pipelines they're using.
Absolutely. It's definitely a very big project with a lot of participants, so
Yeah.
a lot of moving parts for sure. Absolutely. Next question I have, are any targeted metabolites measured?
I think so. The projects that go into the Million Microbiome of Human project are also the projects that we are sequencing in-house. Some of them we do also metabolite measurements. Different ones depending on the study question. They are, however, not part of the Million Microbiome of Human project. Another thing that we want to focus on in this project is to facilitate communication between research groups that are working in this field. If there are metabolites measured, they will not go into the database. At least that's not the plan for now. However, the groups that are participating can obviously exchange all the information that they want to exchange, and we hope to just build a bridge between all those participating groups and make it easier for them to share data between each other, not only the data that we're producing for that project or that goes into the project, but also other data, and potentially start new projects together, bigger projects together, accumulate the data into new projects. There are metabolites measured, but they're not part of the project itself.
Absolutely, absolutely. Thank you. All right, next question. I'm going to ask this one. What steps has the MMHI project taken to comply with access and benefit sharing obligations under the Nagoya Protocol?
I'm sorry, I'm the wrong person to ask for this. Also, that would also be the group in Copenhagen. Maybe if Binyam wants to answer this or can answer this, he can hop in. But I'm unfortunately not able to answer this question.
Understood. All right, thank you for all the questions that are coming in. I'm going to move to the next one. Thanks for a great presentation. So this is a question related to data, and it's possible we may have already addressed it, but I'll ask all the same. Is it possible to share some data with academic researchers to use for their own purposes?
That's an interesting question. So as I said before, if you're interested in any of the projects that you have been that you have seen, I don't know exactly what you're referring to with some of the data. But if you're interested in any of the projects that you have seen, you can get in contact with us regarding your specific question, and we can talk about a collaboration. We are super, super happy, I think, as individual research groups and also as MMHP, as Research Consortium, to go into new collaborations. So the data that is going into MMHP is, as I said in the beginning, going to be restricted to the participants of MMHP. And only later open for public access. But if you're interested in an academic research collaboration, please get in contact either with me or with Mathieu, or check out the information at the webpage of the Million Microbiome of Human Project and get in contact with the coordinators that will give you the right contact person to ask.
Absolutely, absolutely. And then this question I have here, well, it's phrased more as a statement, but I'll ask it as a question. I was wondering if you could provide any insights related to validation that's being done for the study.
Yes. Again, this is a very, very broad question. We do validation of everything. We have validated every single part of the pipeline. We have validated all the sample taking procedures and all the sample material. We have validated the extraction procedures, the library preparation procedures, and the sequencing procedures in all the individual labs with different standards that we use externally and internally. That means we use them as separate validation samples, but we also use them within each individual sample to make sure that the sample itself is processing as it should under the conditions that we are actually treating it. If you use external samples, you always have a bit of a bias that comes from the nature of a validation sample. They're often very simple and they're not treated within the samples. You want to make sure that your sample is right, which is why we also use internal standards that are spiked into the samples. We have been doing, and we are currently doing, a new round of validation between all the labs. So we make sure that all the processing labs within MMHP have the same processing standards to make sure that we have as little bias as possible between the processing labs of MMHP. And that if there are biases, we can define them exactly and say, this bias, this lab produces samples that look a bit more like this. I think in total, it's not unavoidable to have slight differences between different processing sites, but it's important that you are able to validate them. And we have spent, I think, at least the first 5 years of the 10 years that we have been doing high throughput sequencing now, only with validation and optimization of the pipeline. If you're more interested in the exact procedures for every step, please get in contact with me. We as a company, we also do offer services to help you with internal validation processes and guide you through building up a new pipeline or building up a new lab.
Excellent, excellent. All right, next question I have. Do you provide any training in data analysis?
We as a group at the Center for Translational Microbiome Research do not for external researchers, only for collaborating researchers.
Wonderful, wonderful. Okay, I think that we were able to get through some good questions from our audience today. Thank you so much, Micah, for taking the time to answer questions.
Thanks for the great questions. They were awesome.
Yes, absolutely. It definitely seems like the audience learned a lot from the presentations, which is what we want, and that is awesome. That is all the time we have for today's webinar. As mentioned, this webinar will be available to view on demand very shortly for repeat viewing or to share with your colleagues who might not have been able to tune in today. So please keep an eye on your email. You should get a link within the next 24 to 48 hours that will allow you to access both the presentations and this Q&A session. Many thanks again to Matthew and Micah for their presentations, and thank you, Micah, for being here to answer our questions today.
It was my pleasure. Thank you.
Absolutely. I would also like to thank our sponsor for today's event, MGI. Thank you so much. And I would also like to shout out the superb Gen Multimedia Group for all their work in hosting and putting this together. So thank you, Liana Jabs, David Mosley, and Hannah Turner. And most of all, thank you so much to you, our audience, for being here today and for asking such great questions. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.